Good morning, my name is Dr. Chris Pantasis and I'm here to do my first unboxing video of the eScience Lab Kit. The eScience Lab Kit, it's alright. The eScience Lab Kit is what you should have gotten in the mail. You purchased it from the bookstore. It's not included with the cost of the class, so you need to make sure you have one of these. You will also need, not just on top of this lab kit, you will also need your lab book. So you're going to purchase a virtual lab book from the bookstore. Please make sure you've done that now. But we're going to open up the box. To open up this box, you come to the front of it, and it swings open very carefully because you don't want to rip it. Because if you rip it, the box falls apart, and you can't use it to store all this stuff for the rest of the semester. And you will need it for the rest of the semester. So keep track of everything. A lot of fun stuff in here. It's a look at this, completely packed box. Not only is it completely packed up, it was so completely packed that after we opened it before, I pulled something out and it didn't fit back in. So there's a lot that we've got going on here. Let's go through everything in this box. Luckily, on Canvas, you have a handy dandy list of everything that should be in this box. Look for this list. Check through the list, going through everything in the box like I'm about to do. So first off, it says, item number 1, safety supplies, gloves, table spread, goggles, and apron. That's what's going to be in this little bag here, the one labeled safety kit. Go ahead and open it up. Let's see what's inside. We got super stylish safety glasses. These are good for putting on. Obviously, they protect your eyes from spills. They also make you look great. They help you attract a mate. We get into that in Bio 102. We have, check this out. Oh, wait, 2 things? That's amazing. One of them is an apron. Be careful with the apron because when you're putting the apron on, it's very, very easy to rip it. You don't need to wear it while you're unpacking this, but I like to wear things in general because it's fun to put on stuff. So you gotta be very careful opening it up. You don't want to rip it. If you rip it, it's no use to you. It's almost like a garbage bag. Almost open, got it. You've got part you can put over your head. If your head fits, that's great. My head fit. You can tie it in the back, and then you look super cool. You will be the envy of all of your friends. Not only do you have this nice safety apron here, you also get a tablecloth. This is going to be important. It's not a cloth, really. It's it's to protect your table because we will be working with some, they're in here, some caustic chemicals. These are meant to be disposed of. So you lay it out in front of you. Yep, you can see it in front of me right there. This is going to be where you do your, this is where you're going to be able to do your experiments right in front of you. Oh, you know what? I'm going to ask some specialized advice. Which way would you recommend that they set this up when they're doing their experiments? Cloth side to absorb, plastic side to catch.
Actually, plastic side.
Plastic side to catch? I think that's a good idea. So this way, if you spill, it catches it here. It doesn't get it on any of your table.
Sometimes they'll also have their computers next
right. That's a great point. You'll usually be doing your lab with your computer set up with you. So be careful about that. All right, I'm just going to set these aside and see what else is in the safety bag. It's awesome. Look at this. I'm going to say it's gloves. I'm hoping it's gloves, otherwise it's a hairnet. Nope, definitely gloves. With any luck, these gloves will fit you. They are nitrile gloves, which means that they are good for people who even have latex allergies. So you've got a few sets of gloves you can use for these experiments. You don't need to save them between experiments because you could have contaminated them, got something on them, and you don't want to hurt yourself later by putting on a used set of gloves. So there we go. I'm going to put the gloves back. In this particular bag, there's another set of safety goggles, but I think you guys only get one set. Is there anything else in here? And some leftover plastic trash. So that's what's in your safety bag. So item 1, check. I'm going to take a pen and just check off that that's there. Go team. Next up, it says we need a graduated measuring cylinder, plastic, 100 milliliters. plastic 100 milliliter graduated cylinder. Now, Most graduated cylinders will have a wider base at the bottom, so you need to be really careful when you're using this. Throughout the lab, you're going to be measuring using the gradations on the graduated cylinder. Be careful when you put it down. Also, just double check to make sure there's a bottom to it, because if not, you pour liquid in and it goes straight down, and then comedy occurs. Unless it's an acid or a base, at which point death could occur, so be careful. So that's your graduated cylinder. I'm gonna go ahead and check that one off. Next, it calls for a beaker, plastic, 600 milliliters. What's in here? Why? It's a plastic beaker. And is it 600 milliliters? Yes, yes it is. And you can read that right along the side. So I have a 600 milliliter plastic beaker. This is going to be very useful for transferring and measuring large amounts of liquids. Now that I've gotten that one there, I'm going to go ahead and check it off. Moving on to a Petri dish, or like my advisor would always say, it's a Petri plate. Why'd they call it a Petri plate? Because they were Canadian and that's what they did. So let's see if we can find that Petri plate or Petri plate buried in here somewhere. You see it? Oh no, now we're all looking for it. There it is, I found it, it's off to the side. There's a lot of them. Check this out. Yeah, there's a lot. You probably only have one.
Just show one maybe.
All right, so this is what it looks like. It looks like transparent. You can see right through it. It's a small dish. It's a small plastic dish. It's got a top that fits right on top of it. Make sure it has the top. If it doesn't have the top, you've got to contact us. We'll make sure that you can get one. You do need both a top and a bottom for this. It's useful for several different experiments and for containing small amounts of liquid. So I've got my Petri dish. Large glass test tubes. It does say glass, and it says no lip. These look pretty big.
6.
Well, we're on.
There should be 6.
There's 6? Oh, okay.
Petri dish.
6 Petri dishes?
Yeah.
You know what? I got 6 Petri dishes right here. They're in bubble wrap because they do break very easily. You don't have to keep them all in the bubble wrap, but you got 6 Petri dishes in this container. They look like that. Over to the test tubes. All right, so that says lipless. 6 large glass, lipless, gentle test tubes. Right there. They're very, very wide, very transparent. You're gonna have to use these. Now you'll notice if I was to set it on the table, it's gonna fall all over the place. Eventually in this kit, I'm willing to bet, we've got a test tube rack. So let's keep looking and see what we have. But I have my 6 large glass test tubes. Come over to the side. My little science table. You know what? When we got large test tubes, we may as well have small test tubes. That's what's next. And we've got small plastic test tubes with no lip. These small? Those are pretty big. These look small. Yeah, there we go. Small plastic test tubes. Much, much smaller than the large plastic test tubes. Look how tiny those are. This little cute little test tube there. It's like the little brother of these guys. So make sure when you're doing your labs, you're using the proper test tube. You've got your small plastic test tubes here. There should be 6 of them. Let's seal that. No science experiment would be complete without a thermometer. So right in here is a thermometer. It should be wrapped up really nicely because if it falls out on the floor and breaks, be careful with the thermometer. These are ethanol thermometers. They're not mercury thermometers, so there's no dangerous materials in them. With the thermometer, they do come with a case to keep them protected. Try and keep it in that case. This is used for measuring temperature. This one is actually, it is only in Celsius. So for all of you American Fahrenheit fans out there, you're out of luck. We deal with Celsius because we do science. Please note with the thermometer, and I'm sorry that I have to tell you this, I know you're college students, I know you know this. It's the red end that goes in the liquid. That's important. Red end in the liquid, that's what tells you the temperature. Put it back in. Well, just set that aside for now. Moving along. Check mark on the thermometer. Moving our way down here. We've got a plastic transfer pipettes. 5 milliliter. let's see here. There's a all right, so 5 mil. These is a plastic transfer pipette. You can see that it's got 5 marks on it. 1, 2, 3, 4, 5. Those are for milliliters. In order to use these, you squeeze the bulb, put it in the liquid, release the bulb, liquid gets sucked up, and then you're going to dispense however many milliliters as you need from this tube. Important to note when you're using these, just because I don't know when the next time I'll have you is, don't put it in the liquid and then blow bubbles. Make sure you squeeze the air out first and then put it in the liquid. Otherwise, you're going to you could disturb the reaction, or if you're dealing with microorganisms, they really don't like it. So there we go. We got a whole bunch of plastic transfer pipettes that are 5 milliliters. Check mark. I'm just surrounding myself with science. We also have, that's the same size, where are the 1 milliliter transfer pipettes? Those aren't 1 mils, are they? Some of them are. Let me pull it out. So I got a hodgepodge in here. You shouldn't have that. There we go. Nope, that's still not a 1 milliliter. Okay, I'll keep looking. Could be that this kit is missing its 1 milliliter one, so I will see. Yeah, about finding a 1 milliliter pipette. So I can't cross that off my list now. It wasn't there. This is something I may need help with. So I'm going to just put a little circle next to it to say I need to find it. pH paper strips. When we're dealing with pH or the power of hydrogen, you're going to evaluate it based on the color of these paper strips. So I'm looking for the paper strips. They're somewhere in here. Man, going through everything. There we go. Look at that right there. I'm going to come right up to this camera on this one. Behold, the paper pH strips. You've got colors right along there. Those colors are going to tell you what, once you've dropped a liquid onto these papers or dipped the paper into a liquid, it will tell you how much hydrogen is present in relative values in that liquid based on the color. So make sure you have this pH strip test. We got that one, unlike those test tubes, or those pipettes. Let's cross that off. Dialysis tubing. So we use dialysis tubing, which is literally the same stuff they use for dialysis for your kidneys, in order to test osmosis and diffusion later in the course. So let's see if I can find some dialysis tubing. Is this dialysis tubing? No, that's a big plastic container. Let's see. No, that's not dialysis. What is that? Figure that out in a few minutes. Got other stuff. I don't even know what this is. There we go. Dialysis tubing. It's hard to the reason I missed the dialysis tubing, I'll be honest, is because it's really, really hard to see. Obviously, I'm holding up a little plastic bag here, but inside the plastic bag, I don't even know if you can see it from there. You've got a tube. That tube comes unwound, and it is utterly transparent. This will be super, super easy to lose, but we've got it. So go ahead and put your dialysis tubing back in the container. We have our 1 dialysis tube, and that is all we need for the whole semester. I'm going to set that aside and put a check mark. Next, we're asked if we have 10 milliliter syringes. A syringe is like what you get in the doctor's office, right? You always think it's a needle. The syringe is not the needle. The syringe is the plastic part with the plunger at the top. So let's see if I can find, yep, there they are, right there. We've got 10 milliliter syringes. Let me open it up. So we've got, for our syringes, you'll see that they have 10 milliliters right up along the side. You pull it down, you push it up. Basically, you're going to use these to move liquids. I believe we're going to be using them for the photosynthesis experiment. So, you've got your syringes. How many syringes? 3. 3 syringes is what you got. I'm going to set that aside. Guys, and I will tell you, so while we were talking, one of my colleagues just went and found our plastic transfer pipettes, 1 milliliter. So please, 1 milliliter transfer pipettes right there. You'll notice that they are much, much, much thinner than the 5 milliliter pipettes. Make sure to use the proper one. You are so good at this game. Let me get up there. Come up here. Hello. Look at the difference in size there. One's really thick. This is the 5 milliliter. One's really thin. That's the 1 milliliter. Make sure you know the difference and you're using the proper one when you're doing your research. Thank you. All right, so now I can put a check mark that we've found them after that plastic transfer pipette. If you do not have an item in your kit, please let your instructor know so that we can take care of that. Yeast packets. What? Probably missing?
Because it's used.
Yeah, all right, so we don't have any yeast packets in this one because they have already been used. Yours should have a small packet that's roughly 2 inches by 2 inches that says active yeast on it. So we'll look to see if we can find another one, but for now I'm going to go ahead and put a 0 next to it, a circle, so I know that it wasn't in my lab kit. A plastic funnel? Oh, I love funnels. Funnels are a great way of moving liquids from one place to another. Oh, I'm disappointed. Now I know what this is. Oh, that's a coffee filter. I am looking for the funnel, but I don't see a funnel anywhere. This isn't the funnel, is it? I don't even know what that is. We'll get to that soon. So we need a funnel, a plastic funnel and a packet of yeast, and that's what we're missing at the moment. So I'm going to go ahead and put a circle on my plastic funnel. This is like a real unboxing. A metric ruler. Guys, I was wrong. I found the plastic funnel. Yeah, so the plastic funnel was in here. It's just at the bottom, and I was looking for a funnel, and I thought it was a beaker. So this is what the plastic funnel looks like. Again, college students. Let me get right up here so I can tell you closely. You are college students, you should know this. When you're pouring into a funnel, you pour from the big end through the little end into your test tubes. You do not invert it and try to pour from the top end. These are problems I have seen and had to deal with. Don't make this happen at your house. Liquid's in the top end. And while I'm here, we have our active yeast. This is what's going to come in your lab kit. It is, so active dry yeast. You're going to activate this when you do the lab, in the manual the lab says, with warm water. Not with hot, with warm. So we got our yeast, we got our funnel. And, nice, we have a metric ruler. So with your rulers, you're going to see a few things. One, you're going to see some large parts at the top. Get in there. Large parts at the top that are going to show inches. It shouldn't even be labeled inches. We're not going to use that. You're going to use this other side that's labeled in, oops, there we go. The other side that's labeled in centimeters or cm. The large tick marks, 1, 2, 3, 4, and so on, are centimeters. The small tick marks are millimeters. Make sure you are following what the lab says for the measurement. I guarantee you it's not going to say inches. It's either going to say millimeters, the small marks, or centimeters, the large marks. So please be careful. Great, so we found a few other things that we're missing. We've got that plastic funnel, check mark. We've got the yeast packet now, check mark. We've got the metric ruler, and we're halfway through our list. Guys, this is the thing that throws everybody off. There is in your kit, it looks like a flat piece of cardboard. It is actually your test tube rack. It's got holes in which you can put your test tubes in. Let me grab a test tube and show you. Here we go. Test tube, holes, goes in, and now it stands up by itself. Do not throw, oops, there we go. Do not throw this test tube rack away. A lot of people accidentally throw it away thinking it's just extra packaging. It's like an origami project. You fold it up and boom, you've got a test tube holder. So, test tube. You want to do a demo?
No.
Okay.
But basically,
Come on in.
okay, so what I want to tell you guys is when you're sending your selfies, where you show us the color of the reaction, hold your test tube rack like that so that we can see the colors of your solution. Do you see? Many of you just send me a picture like that, but do you see this is covering the solution? So keep it like this and take a picture like that. If possible, keep a blank sheet in the back so that we can see the colors of your test tubes, okay?
That is the best way of recording your data, because guys, you will have to record all of the data you send us. You need to make sure that we know that you did this experiment on your own and you got the results that you probably were expecting. The reason for that is, even though with science, we make mistakes all the time, experiments don't work, but what we want to do is make sure that you are following directions. So here's our test tube rack. Don't throw it away. Take the pictures from the proper side and make sure that you're careful with it. Test tube rack, check. So I have on this list that there are 2 plastic funnels. I believe there's only supposed to be 1, so we're going to update this list.
Yeah.
Just cross that off. Pinto beans.
No, they don't need it.
You don't need pinto beans?
No.
Pinto beans don't need either. Man, this is going quick.
Yeah.
Cheesecloth I do know we need.
Yeah.
And I got some cheesecloth right here. So cheesecloth is, the reason it's called cheesecloth, history lesson, is when you're making cheeses, oftentimes you're going to need to squeeze out the whey, which is the wet part. So basically a cheesecloth is just a piece of cloth that's got lots and lots of holes through it. These holes are useful for straining. So you don't need to rewash this or try and keep this when you're done, just get rid of it. We're going to use this for straining in our lab. So that's the cheesecloth. Go ahead and put that away. It's also used in a lot of crafts. People make ghosts out of them for Halloween. I know. So, you know, if you ever get tired of biology, Halloween decorations. What else do we have? So cheesecloth, check. Coffee filters. Guys, it's not because we're trying to keep you awake during our lectures or during your readings. You're going to need coffee lectures for your actual labs. Coffee filters, for those of you who don't drink coffee because it'll stunt your growth, or so the legends say, basically look like this. Look, they were all folded up into a nice triangle, and you unfold them to a circle. Coffee filters are also used for filtering materials. A little bit finer of a filter than the cheesecloth. So be careful with this. You're only going to use it 1 time. And then apparently now I've got to fold it back up in such a way that it goes back in the container. So that's your coffee filter. Get in there. Over to the side. We already looked at that. Check mark on coffee filters. And then there's just a big list that says chemicals. So luckily we've got our chemicals over here. We've got a long list of them. Let's start working through them one by one. I gotta be very careful with these. In fact, I should be wearing gloves with these, so let me go ahead and grab those.
Do you want me to give you one?
No, I got them. I'm gonna see if I can do this without being handed them. I'm gonna use some gloves, because when you're handling unknown chemicals, you don't necessarily know everything about these chemicals all the time. So I'm pulling out a pair of gloves to handle them. And I don't know if in shipment, any of the containers have cracked or shattered or anything, so we want to be very careful. Yep, small gloves. Awesome. I hopefully will still have blood flow at the end of this. There we go. Get another one in. Oh, 2 fingers went into the same slot. That's, come on. Putting on gloves, definitely a fun activity. Look at that. All right, I'm gonna go through these. So we've got, this is going to be where I bromothiamyl blue. This is a stain, so be very careful whenever you're using a stain, because as the name implies, it will stain you. Your hands will turn Smurf blue. It's not a fun thing. Your clothes will turn Smurf blue and then stay Smurf blue. So bromothiamyl blue. Check mark on that one. We've got, what else do we have in here? Going through one by one. We have glucose. Glucose is simple sugar water. We're going to learn a lot about glucose this semester. So glucose is not terribly dangerous, but even though it's just sugar water, don't drink it. Glucose, check. We've got ethanol. Ethanol is alcohol. Don't drink it. The end. It is flammable, so be careful. So ethanol. Check. 95%. This is easy. I got this. Lactose. Lactose is another sugar. We're going to be using sugars later to test, actually we're going to be using sugar with those yeasts to see what they like to eat the most. So this is the lactose solution. It's shelf stable. Again, do not drink it. Lactose, check. More glucose. I already checked that one off the list. Green food coloring. With green food coloring, it is not dangerous. You still don't want to get it on your clothes. This is going to be very useful. The food coloring is useful for making it so that we can see what's happening in your reactions. So food coloring, check. Oh, one color each. Guys, I marked off food coloring. I can't mark off food coloring yet. I can only mark off that I have green food coloring. Let's see what else we've got. It's yellow food coloring, which looks red, but it's not. It's yellow. We've got red food coloring. We've got blue food coloring. And then based on this, I am missing iodine, maltose, and starch, which I'm betting are in this other container. I'm gonna keep going. Let's see here. Vegetable oil, that's not on this list. Should it be on the list? No, that's just old. I'm gonna get rid of that. Put it over here. Nobody will see that ever again. Let's see what we got here. Starch. Now, starch is on the list. So again, starch is a solution that we don't want to drink. It's not terribly dangerous. It's one of our macromolecules we'll discuss later that contains, go figure, starch. So starch I can check off. Got my food coloring. I'm looking for iodine, which should be, it's the wrong lab. No, bio 102. Also bio 102. Clove. It looks like I am missing iodine, maltose, and bioarray solution. So what do we got here? Oh, great. Iodine, it's going to come in this dark container because it is photoreactive. That means it's going to be impacted by light. So you're going to have iodine, check mark. Maltose solution. Maltose is another sugar, so not terribly dangerous. We're going to be using this with our yeast. Just make sure it stays sealed or else you might get some bacteria in there or fungus that really love to eat it. And bioarray, yes. Bioarray solution is the very last one. It's another one of these blue solutions. Bioarray solutions, one of the more dangerous solutions we've got because it's going to contain sodium hydroxide, which is a strong base, so be very careful with it. It's also one that is very commonly disposed of at the end of the, when you're finished with your labs. So, in this case, bioarray is a deep blue solution that will turn purple in the presence of proteins. And if you really like to spit at people, make sure you say purple in the presence of proteins, because you can't help yourself but do it. So there we go, bioarray solution, check mark. That is everything in the kits. Again, if you are missing anything in this kit, please make sure to let your instructor know so we can get that fixed. Now I've got the super fun task of putting all this away that I won't make you watch. If you have any questions or concerns, please contact your instructor. Have a wonderful day.